We have one final speaker today. His name is Dr. Jeffrey Staudinger. He's very well known on this campus. He'll be presenting on genomic insights into drug-mediated metabolic pathway modulation in colon cancer cell line. Say that 3 times fast, right? Dr. Staudinger serves as the director of the MSSU-KCU Stress Research Consortium. The MKRC is housed within the Biomedical Research Laboratory, which is comprised of about 3,000 square feet of office and wet lab space located on the campus of Missouri Southern State University. He also serves as the course director for the Biomedical Science Research Elective offered through KCU College of Osteopathic Medicine. Dr. Staudinger's recent publications and recent research studies include associations between pregnane X receptor and breast cancer growth and progression, and clinical applications of small molecule inhibitors of pregnane X receptor. His laboratory seeks to understand fundamental regulatory mechanisms in liver toxicology through the identification of novel PXR target drug-inducible genes in tissues throughout the body. Additionally, his team seeks to unravel the network of signal transduction pathways that interfere with PXR, mediating drug metabolism and drug transport. Let's give a very warm welcome to Dr. Jeffrey Staudinger.
Step. Thank you, Vicki. Hello, everybody. Hello, students. It's nice to see you. Good to be with you today. I'd like to, first and foremost, thank the organizers for inviting us to this presentation. I personally am super excited to do this for a myriad of reasons. In particular, I'd like to thank Dr. Van Galen for providing the laboratory space in which KCU and MSSU can collaborate on this campus. We're really starting to get some momentum up and running, and I think some of the data that I'll show will prove some of that to you. So briefly, as director of the MKRC, MSSU-KCU Research Consortium, our goal is to build research opportunities for MSSU students in collaboration with KCU student doctors. In particular, the MKEEP program, administered by Dr. Victoria Rhodes, has been key in our growth over these last few years, so I appreciate you as well, Dr. Rhodes. There's some hot links. I think eventually these slides will be available to you and you can learn more about me. But briefly, I want to show you where my passion for science comes from. Like our previous speaker, I'm a Nebraska product, a Lincoln, Nebraska product, where my father was chair of biology at Nebraska Wesleyan University for like 20 years. So as a young child, I grew up in the lab, basically learning to bend glassware and not start the lab on fire with Bunsen burners. So I have some knowledge on how to operate an autoclave as a 6-year-old. And so that was fun. That's initially where my passion came from. And his tests, which I had 2 courses where my father directed, were so easy. I could imply, oh, that's the answer, because I knew his sense of humor. It was ridiculous. Okay. Eventually, I got a PhD in biomedical sciences, and I know you offer a degree in biomedical science here as an undergrad, so with an emphasis in biochemistry and molecular biology. So I've been described as a molecular endocrinologist. I did a postdoc at GlaxoSmithKline, Breakthrough Medicines for Everyday Living. Postdoctoral fellowship at KU Med Center, where I refined my research interests as an adult. And then assistant associate full professor at KU for climbed the ladder for 16 years. And then I learned, wow, they're opening a brand new medical school in Joplin. And so I knew the difference between a state-funded institution and a private institution. And I thought, this is going to be a great challenge for a guy like me to help open the medical school. So in 2017, my wife, Dina, and I moved to Joplin, and I have not regretted it for a moment. It's been an adventure, and we are so happy to be here as a professor at Kansas City University. So that's who I am. Now I'd like to tell you a little bit more about who we at the MKRC are and what we can do for MSSU students. MKRC, let's talk about MSSU briefly. They've been serving this region since 1937, a long-established tradition of excellence that I'm just so excited to see the Wilcoxon Summit moving forward and the things it's going to bring, and the emphasis on health care related education, I think, is a key strategic decision, and I fully support that. So in 2017, KCU opened the first med school in nearly 50 years in Joplin. And as all of you know in this room, Joplin is the center of healthcare access in the 4 states region. So it was my bright idea to found this MKRC, and with the express purpose of identifying new health treatment opportunities for this community. As the anchoring location for the MKRC, and we call it the BMRL. We support basic and clinical research that investigates complementary and integrative healthcare approaches. Okay, so that's what the MKRC is. Who are we? With me today is Mr. Dan Brobst, and Dr. Creamer, some of you will know, former chair of biology at MSSU, got recruited away to KCU. He and I are key collaborators in this endeavor, and we occupy space in the building right across the street, the Justice Center, Mills Anderson Justice Center. It's really amazing space. So after 16 years at KU in the School of Pharmacy in Malott Hall, if any of you know KU, I have to say I got an upgrade in terms of the building. So thank you again, Dr. Van Galen, for that. Dan was one of my first graduate students and then morphed into a research associate. So we worked together starting in 2001 for 11 years, and after which he married and moved to Oklahoma City and worked in otolaryngology. But then 14 years later came back to me. So we've got the band back together and we're embarking on new research endeavors. So most of the data I'm going to present here is generated by Dan in collaboration with student doctors and also M-keepers, I call them. The newest addition is Miss Catherine Elliott, a freshly minted master's degree from Dallas. And so that's who we are. Now I'd like to tell you a little bit about what we can do. And basically we've been acquiring a whole lot of new instrumentation. So I'm going to highlight some of that and how we're using that to drive biomedical research in this environment on this campus. We can do real-time PCR. We have both 96-well and 384-well PCR machines. The 384-well machine is on the way. We have imaging capabilities. This guy can do all sorts of things, not just Western blots, but it can image cells and fluorescence and UV and visible light. It's a fairly interesting and enlightening piece of machinery. Our most recent acquisition is Sci-Fi. So think of a CO2 incubator in which you grow cells and an immunofluorescence microscope that is interfaced with AI and a computer that can take time-lapse images so you can visualize cells in real time and take movies, basically movies, of what a cell is doing through time if you treat it with this or that other thing. So this we're pretty super excited about. Okay, we have a multimodal plate reader for ELISA, for all kinds of assays, any kind of colorimetric assay you can think of. These are some of the capabilities that we brought to this campus. And now I'd like to introduce the most recent acquisition that we have validated and we have up and running. And a lot of the data I'm about to show you was generated on this next machine. It's called Illumina NextSeq 1000. And again, it's on your campus just across the street. I encourage you to come visit. What can this thing do? It's amazing. Our first speaker, our keynote speaker, talked a little bit about next generation sequencing. So historically, the first human genome was released in the 2000s, as she mentioned, but the cost was around 2.7 billion, and it took a decade to accomplish. What can this machine do? Well, it's $1,000, and I could sequence maybe this half of the room, or this third, this section, all at once, and get some fairly interesting data on each of you. So that's how far next generation sequencing has come, and this technology lives on your campus. So I think that's pretty interesting. Okay, now that I've told you what we can do and how cool it is, I'm going to tell you my passion. There's something that has plagued me since I was a small child for whatever reason, and that is, in Greek mythology, this is an analogy, Prometheus got punished by Zeus because that rascal gave fire to the humans, and Zeus didn't like that, so what did he do? He chained him to a rock and sent an eagle to eat his liver every day. So the question that has plagued me is, how does that liver regenerate? How do our genes and our environment interact? I think this is where the real action in science lies, gene-environment interactions. So part of the answer is the nuclear receptor superfamily of ligand-activated transcription factors. So students, I'm about to embark on a biochemistry lecture. So take some notes. There will be a quiz, and there's extra credit involved as well. Right? Okay, from the pharmacology perspective, there's an interesting slide here, and that is, of all the drugs that exist, what % target one gene family or another? So shown here are the rough percentages of drugs that target kinases, 33%. G protein coupled receptors, another 33%. So there's 2/3 of the drugs that clinicians prescribe and pharmacists dispense. Ion channels comprise another 18%, and nuclear receptors, which is the topic of my talk today, are targeted by around 16, depending on who you talk to, 16 to 20% of all drugs prescribed target one or the other family members. So you might ask, what are some of these drugs? So I've made that list for you here. Some of the top 100 drugs that target one or the other of these family members include the glucocorticoid receptor. Clinicians in the audience will recognize dexamethasone as one of the most powerful anti-inflammatory agents on the planet. Also, mineralocorticoid receptor is targeted by certain drugs. Androgen receptor, both testosterone and finasteride is an anti-cancer agent. Estrogen receptor, we heard a little bit about this morning, is targeted by estradiol, as well as tamoxifen, which is a so-called selective estrogen receptor modulator, SERM for short. Incredibly powerful anti-cancer agents for ER/PR positive breast cancer. So this is an antagonist. Progesterone receptor has its own drugs associated with it, and if that's not enough for you, we'll go down the list a little farther. Thyroid hormone is targeted by levothyroxine. I'll talk a little bit about PPAR gamma later in this talk, which is a new critter to me, but I have made some inroads into its biology as well. These receptors are targeted by thiazolidinediones as well as lipid-lowering fibrate drugs. These drugs are highly used in diabetic individuals with some success, although some of the more recent biologics are taking control, GLP-1, GIP agonists. And last but not least, treatments for acne, retinoids. Okay, so this is a clinically relevant area of study, is the point. And from a fiduciary standpoint, it's also incredibly important. Annual sales exceed 30 billion, and this year alone, nearly $90 billion in drugs will be prescribed through one or the other of these family members. Okay. So what is a nuclear receptor? Here's a cartoon. It's hard to do in two dimensions, but I'll do my best. So a ligand in this case, and by the way, the best drugs are greasy. They're lipophilic. They need to be converted to more water-soluble compounds to get out. I'll talk more about that in a little bit. But the ligand associates with Pac-Man, the red Pac-Man, and forms a complex. That complex then turns into a square and binds to DNA in the nucleus where it regulates gene expression in those target cells where it's expressed. You'll recognize some of these hormones. They look a lot like cholesterol, a lot of them. So for example, estradiol binds the estrogen, red estrogen receptor. It goes to the nucleus in tissues such as breast and uterus where the estrogen receptor is expressed. Take a minute and think about that. That's specificity. One of the biggest challenges of drugs is off-target effects, but nuclear receptors, by virtue of their expression profile, have the biological effect of the ligand only in those tissues. So they're very druggable is the point. Cortisol, vitamin D binds the vitamin D receptor, progesterone binds the progesterone receptor, so on and so forth. Now, what does it bind to, this complex? Well, it binds to DNA and the promoters of genes that they regulate. And the core sequence for these genes is AGGTCA. So those of you who've had intro to biology, frog biology, you know DNA is composed of 4 chemicals, A, C, G, and T. So these receptors have an affinity for that sequence. Okay, that's what they bind to. And here's another two-dimensional picture of how the whole thing happens in my mind. So PXR, which I'll talk a lot about, stands for pregnatin X receptor, expressed in the liver and intestine at high levels, also in certain cancers, which is the topic of the data that I'll present. Associates with histone deacetylase, HDACs. And in the absence of ligand, there's NCOR and SMERT, which deacetylate histones. Now, they taught you about beads on a string, right? And unwinding beads on the string. When you deacetylate histones, the beads are really tight on the string. And in gene expression, it's all about access. So when you add the ligand, there's a conformational shift that I showed you in the Pac-Man cartoon that tends to change the shape such that the dissociation of co-repressors is favored, while as the recruitment of co-activators, so-called histone acetyltransferase proteins, acetylate histones, the beads fall off a string, the chromatin unwinds, and gene expression ensues. So this is the molecular mechanism of how nuclear receptors generally work. Okay, let's look at the whole superfamily. They call it a superfamily because there's a bunch of them. It's the largest gene transcription factor superfamily in metazoans, and it's an amazing superfamily. But they all have the same conserved overall structure: an N-terminus with an activation function number 1, a DNA binding domain. It's a zinc finger, so it uses zinc to intercalate its fingers into DNA physically. A ligand binding domain, which is where the ligand binds, and then AF2 is a ligand-dependent activation function. Now, in the history of endocrinology, it went like this. A lot of these hormones that are listed here were identified, some in pregnant female rabbit urine, progesterones in particular, and some of the mineralocorticoids. That eventually led to the cloning of the first nuclear receptor, glucocorticoid receptor. It's a debate. I say Keith Yamamoto in the University of California, San Francisco. Others followed very quickly once they determined the protein structure. So this was before molecular biology. Okay, then along came molecular biology, and they cloned all these orphans after thinking, aha, we know all the hormones and we know all the receptors. But then they had these techniques to identify using low stringency hybridization, degenerate PCR, fancy things. And they found all these orphans. They call them orphans because there's no hormone associated with them at the time, but they had the conserved overall structure. And so the question then became, oh my gosh, these are all druggable targets. What are these other guys doing? So that's kind of where I came into the field as a postdoc at GlaxoSmithKline. They had the audacious goal of cloning and characterizing receptors from rat, mouse, and human, all of them, and identifying all ligands in all species for all of those receptors. So my job at the time as a postdoctoral fellow was to use the Genome Project to identify any as yet to be identified nuclear receptor superfamily members, and we did that through the Human Genome Project, and adopt the orphans. In other words, find the ligands that activate these guys. And today I'm going to talk to you about two in particular, PPAR gamma and PXR, pregnane X receptor. Okay, so that sets the stage for what comes next. So this was, I don't even know what year it was. It was around 1998, when PXR appeared as a sequence in the Human Genome Database. And immediately, our computers were trained on the Human Genome Project. Aha, a novel nuclear receptor. So the very first thing I did was clone it. And it turns out in a western or northern blot, it's highly expressed in liver and intestine. So we're about to take a journey into the enterohepatic system and follow a drug through its path of oral administration through your gut and intestine. So the nuclear receptors in the enterohepatic system are key in regulating lipid, drug metabolism, drug transport, as well as glucose homeostasis, bile acid homeostasis, and oxidative stress. So these are very important players in the health of your GI system, and they interface with cell-signaling pathways in pathological conditions to include inflammation, diabetes, and obesity. So let's get a little deeper, a little closer. By way of description, I want to tell you the last 20 plus years of my career can be summarized right here. What are the ligands that activate PXR? And what are the target genes that it regulates? You know, at the time, all I had was a sequence of DNA and a novel nuclear receptor and a northern blot showing high expression in liver and intestine. But some of the drug screening we did at the drug company turned out pharmacological agents like rifampicin, a drug I'm going to talk about, SR12813, which is a failed drug candidate, St. John's wort, herbal remedies activated, and many, many more. It turns out hundreds, if not thousands of compounds, some of which we don't even know yet, can activate this receptor. So you can think of it as a broad selectivity receptor that regulates the expression and activity of very important genes involved in drug metabolism, including cytochrome P450s, which do oxidative metabolism, among other reactions, UDP glucuronyl transferases, sulfotransferases, carboxyl esterases are all regulated in a drug-inducible manner by this receptor, as well as drug transporter proteins to include breast cancer resistance protein, MDR1, the drug efflux transporter, OATP2, an uptake transporter, so MRP3, MRP2, again, drug efflux transporters. So it appears that this critter, pregnane X receptor, regulates the drug-inducible gene expression in liver and intestine of our protective mechanisms. Mother Nature is so tricky. She gave us all these tools to deal with the onslaught of things that we're going to be exposed to in our environment. This spoke volumes to me because of my curiosity about gene-environment interactions. So this is the mechanism whereby our liver and intestine induces the expression of these very important genes that regulate drug metabolism, drug transport. Okay, so let's get into phase one a little bit. This is where the biochemistry lecture starts. And by the way, here's some assigned reading. It's a great article called Human Cytochromes P450 in Health and Disease, published in 2013. This is a clickable link. If we get the slides to you, you should be able to click on a number of links. What I'd like to say about this family right away is it's the most broadly distributed gene family in the known universe. Many people who study P450 say evolution occurred because of the advent by Mother Nature of cytochrome P450 genes. In other words, there's CYPs in every living organism in the known universe. It's an incredible family. There's 57 in us. And they do all kinds of reactions. We're going to focus on oxidation. So I know there's some chemists in the audience who, and I'm not one of them, as a biochemist, organic chemistry just about killed me. Are there some organic chemistry lovers in the room? Awesome. Someone's got to love it. Okay, here's how it works. You administer a drug, orally, we're talking. It goes through phase 1 oxidative metabolism, and an OH group gets stuck on it. That OH group serves as a reactive site for glucose addition, called glucuronidation. It can also sometimes serve as a site for sulfonation or even glutathione conjugation. But and then those compounds become substrates for drug transporters. And this is how, every time we take an oral medication that is metabolized this way, by the way, about 50 to 60% of all clinically prescribed drugs are metabolized in this way, get out of the body. So, big picture, it takes a fat-soluble compound, which makes a good drug to get on board your patients, and converts it into a more water-soluble compound to get it out of the body, and your patient has to take another pill the next day. So that's called biotransformation. Okay. CYPs are the first and rate-limiting step in this reaction. Is that clear enough? Everybody understands biotransformation. So cytochrome P450s always occur in conjunction with cytochrome P450 reductase. Now here comes the extra credit question, students. If anybody can tell me where NADPH comes from in our body's biochemistry, you get brownie points. Say it? Krebs cycle, yes, but there's more specifically the pentose phosphate pathway. So you get credit. Well, I'm pretty easy. You get the credit. So it always occurs in combination with cytochrome P450 reductase, which abstracts 2 electrons from NADPH, passes them to heme, so there's iron involved and electrochemistry involved, right? The net effect is to produce water and a hydroxylated more polar metabolite. Now, one drug that this happens to could be birth control, steroid hormones. Steroid hormones look a lot like nuclear receptor ligands, so that's kind of interesting. You get birth control, hydroxylated birth control, and water, and the whole reaction can be summarized generically here. Again, this is for the organic chemists in the audience. And we can still be friends. Okay, so clinically, this is incredibly important because there are inducers of this enzyme, cytochrome P450, shown here listed: carbamazepine, rifampin, I'm going to talk about that drug, phenobarbital, St. John's wort, so herbal remedies have constituents in them that can induce P450s. And this has the effect of increasing the metabolism of things like birth control. So if you're wondering how you got pregnant on the pill, it's probably because another drug or something you were eating induces its metabolism and makes it less effective. This is one mechanism of drug-drug interaction, and PXR is the molecular mechanism that induces P450s. But there is another mechanism of drug-drug interaction, much more clinically relevant when compared with inducers, but nonetheless, inhibitors of P450, as you can imagine, might gum up the works. So if you're on combination therapy with one drug that is particularly toxic and you inhibit its metabolism, it raises the level in your patient's serum and leads to drug-drug interaction. So there's kind of 2 directions drug interactions can go: decrease therapeutic levels of the compound or increase levels of, and if it's a toxic compound, it can have tragic and lethal consequences. So this is all clinically very relevant. Okay, let's talk about CYP3A4 specifically. CYP3A4 comprises 1% of your total liver body weight, but 30% of the total CYP activity in your body. So it's a major player in drug metabolism. Here's a list of some of the drugs that are metabolized by CYP3A4. I don't expect you to be able to read this, but here's a list of some other ones. So you can see that this CYP3A4 enzyme that is highly enriched in our liver and intestine, and this is a partial list. It metabolizes a bunch of things, and they're all very important. So drug interactions become incredibly important. Okay, again, so let's take a two-dimensional cartoon drive through an orally administered drug. So what we're looking at is my cartoon of a nuclear receptor function in enterocytes. So you see BCRP, ASBT, MRP2. The drug comes in through the intestinal lumen and encounters these transporters that are lodged in the membrane of the intestinal lumen, guarding against and regulating what gets into your cells and into your portal circulation. So those drugs are rendered, some of them are substrates, they can get in, and some of them activate these nuclear receptors in the intestine to regulate target genes that might transport such a metabolized drug into the blood, where next stop after the intestine is where students, anatomy students, after the intestine, blood goes to liver. Through the portal circulation, by the way, this represents a therapeutic barrier. It's often referred to as first pass metabolism. So as I said, next stop is the liver. So here it comes through the portal circulation into the liver. Again, it gets taken up into cells where it can regulate the activity of these nuclear receptor proteins, whose job it is to regulate all these genes, whose function is to move things into the bile, but sometimes back into the blood. In the bile, it's excreted in feces, and when it's transported back into the blood by a protein called MRP4, 3, some of these others move stuff back in the blood, it's excreted in urine. So these drug metabolites are handled largely in the intestine and liver and then excreted, and you have to take another pill the next day. Okay, so all of what I told you, this is really my career in the last 20 plus years in a nutshell. We cloned PXR, identified a lot of drugs, foreign compounds, steroids, and bile acids as PXR agonists that upregulate uptake transporters, drug metabolizing enzymes, drug transporting, drug efflux enzyme, and drug transporter proteins. So there's a coordinate regulation of uptake, metabolism, excretion, and efflux. So it's central to drug metabolism. Okay. So the story's kind of been told. PXR is a nuclear receptor, lives in liver and intestine, it does its thing. Great. It was easy to get R01 grants in the early days because it was novel, it was clinically relevant. But the story's kind of been told, and that's how science happens, students, in waves. An opportunity comes, astute people jump on the wave, it crashes, then you have to look for another avenue, another project, another angle. So I started thinking about breast cancer and drug metabolism and chemotherapeutic resistance in breast cancer, and it made sense to me that PXR could be involved. So broadly, what I've told you is that there's environmental stimuli and drug metabolizing enzymes, so-called DMEs, that have an effect, and this is very similar to the slide you showed this morning, Dr. Heine. Herbs and drugs, lifestyle choices, diet, smoking, alcohol, age, hormones, infection, other types of disease, inflammation, all have a huge effect on drug clearance, drug plasma levels, and drug response. And these are all overlaid and influenced by genetic factors as well, gene mutations and the like. Okay, so I'm going to introduce you to two experimental compounds, and then I'm going to get to the data, and we'll wind up this shindig. Two prototypical activators that we use in the lab are rifampicin, and its main clinical uses are listed here. It's an antibiotic. But from my perspective, the most important thing is that it's a well-known and potent inducer of enzyme activities in liver via PXR activation. We know that now. We didn't at the time, which can result in drug interactions, and we've already talked about this. So the second compound I use is called SR12813. It didn't get a fancy name because it failed in drug development due to toxicity concerns. But it was developed as an alternate mechanism to statins. Statins, which inhibit the enzyme active site. Instead of doing that, this drug targets HMG-CoA reductase by promoting its ubiquitination and subsequent degradation. So HMG-CoA reductase levels plummet in animal and cell models that were administered this compound. But when it got into higher mammals, the toxicity came about, so they pulled it from drug development. But it became a well-known activator of PXR, and it's been used now for decades as a model compound to study PXR activity in this model system or that model system. So rifampicin and SR12813, these are the two drugs that we're going to study, and I'm about to present that data. And this paper is a manuscript in preparation. We're in the final stages. Various student doctors and Mr. Brobst and myself are primary authors in this manuscript. Okay, so the question is, what effect does drug treatment have on PXR and CYP3A gene expression in different tumor cell lines? I don't have access to a vivarium, otherwise I do it in animal models, but to be honest, I really don't want to play with mice and rats anymore. They're so cumbersome, and IBC and all that stuff. I was a mouse and a rat farmer for 25 years. I think that's enough. Okay, so let's look across liver, colon, breast cancer that has ER and PR, T47D, so these are hormone receptor positive T47Ds, and triple negative, MDA, MB231s. This is the triple negative breast cancer that is the most aggressive. So it's expression level across cell lines. You can see HepG2, and when I said it at 100, in comparison, colon in LS180 has a significant and detectable level, as does T47D, whereas MDA, virtually undetectable. If there's any there at all, it's not very much. So let's look at its activity. Again, PXR's job is to induce CYP3A4 among other target genes. But in the gray bars, what you'll see is that HepG2, both rifampicin, let's try this, and SR12813. Are you seeing that green pointer? In the gray bars is rifampicin. In the black bars is SR12813. The y-axis is fold induction. The x-axis is, again, liver, colon, hormone positive and triple negative. So in MDAs, likely because there's no PXR, it's not inducible. CYP3A4 gene expression is what we're looking at. Okay, in order to study something, it has to be there. So I'd made a living in liver. I hadn't done much in colon, and I wanted to study cancer. So I thought this is a good model system which I can grow in my lab and study. So we started asking, how does PXR function in colon cancer to regulate drug disposition? So briefly, LS180 colon carcinoma cells are an adenocarcinoma derived from a metastatic site in the small intestine. It's highly used in PK and drug metabolism studies, studies of drug interactions. It has the genes that are known PXR target genes. So briefly, how many of you had genetics, students? Have you had genetics already? No? Yes? Okay, I'm about to give you genetics in the next 20 seconds so you can forget about that course. You ready? Here it goes. DNA goes to mRNA, goes to protein. You're done. No. Transcription and translation. So what we did is we gathered the messenger RNA, and through the magic of molecular biology and reverse transcriptase from retroviruses, we make something called cDNA, or complementary DNA, DNA that's complementary to the message And then we put it on the next generation sequencer. So what we did is we treated with no treatment, with vehicle treatment, which is DMSO, dimethyl sulfoxide, as well as certain concentrations, fixed concentrations of either drug. And then we just sequence everything in mass. And then AI comes along and interrogates a gigabyte worth of A, C's, and G's, and T's and tells us exactly what's going on. So AI is not only revolutionizing the practice of clinical medicine, but biomedical research as well. And I have to tell you, when this thing came into my environment, my spine tingled, I got goosebumps. I still do just talking about it. It's amazing what this thing can do for so little money. Okay, so we did that, and it wasn't Chad, it was some kind of proprietary behind-the-scenes AI. Told us that these two drugs share 53 genes induction upregulated by both rifampicin and SR12813. 53 of them they shared in common. Here's a list of those genes. What you notice in yellow is some of the well-known PXR target genes are in that list. So that told us, okay, it's working. And your facilities people here helped me get this machine online and get a nice HIPAA-FERPA qualified drop so the information is protected. They refer to it as a fancy hot dog machine, which I thought was funny. Anyway, 588 genes are regulated exclusively by SR12813, and there's a much smaller number, regulated exclusively by rifampicin. So that's biology right there. What is SR doing that rifampicin isn't? And I was really interested in that because of some of the other genes that appeared on this list. And so I asked, more appropriately, Dan asked AI, what are the top 30 genes induced by SR12813? And what I noticed right away is there's a lot of really important metabolic genes that are regulated by this compound exclusively, not by rifampicin, that have to do with ketone body metabolism, lipid storage. So these are pathways not known to be regulated by PXR. So I thought right away, SR, especially due to the Venn diagram and this list, is doing stuff off of PXR, so off target, I call it. So what is it? Well, first of all, you generate this mountain of data of ACs and Gs and Ts. You have to follow it up, and that's where real-time PCR comes in. What I want you to notice is, again, rifampicin is in the black bars, SR is in the gray bars. Rifampicin induces the well-known PXR target genes right here, the first two, CYP3A and UGT. We're looking at fold induction of gene expression following drug treatment. Rifampicin induces, as expected, as does SR, same with UGT, but all these others only SR regulated. So we followed up our initial observation, and with two methods, we were pretty convinced that this is an off-target, non-PXR dependent effect. So the question became, if it's not PXR, who is it? So luckily, in my -80, I have an army of these nuclear receptors. I have close to all 48 members as a tool. So we modified a mammalian 2 hybrid system in which we fused all their ligand binding domains, so this is nuclear receptor ligand binding domain, to a generic Gal4 DNA binding domain, which has a reporter gene associated with it, so that when you treat with drugs, you activate this reporter gene. You can see it in one of those plate readers I showed you in the very beginning of the seminar. So we used those nuclear receptors listed here, and I suspected PPR gamma from the beginning because those genes were known to be regulated by PPR gamma. So I needed to confirm that. So when we did the experiment, this is what we saw. So SR12813, and cofactors is just a way to beef up the signal a little bit. So PXR, human, just the ligand binding domain or the full length, works pretty well. Not so much with the mouse form of pregnane X receptor owing to its evolution of the ligand binding pocket. Mice take different drugs than humans do, right? LXR didn't activate, PPR alpha didn't, but PPR gamma, wow, through the roof. So that was kind of the nail in the coffin. But notice this is mouse. PPR gamma. So we built another assay and asked the question, does human work? We also did dose response, and we got respectable EC50s. And then so we moved into a human form because evolution happens, right? Again, PPR gamma activation would activate this reporter gene derived from the firefly. It's just a gene that you can see in the plate reader. And when you do that, indeed, SR12813 activated PPR gamma. So this is kind of coming to the end of that story, and that is to say, we identified new pharmacology for this drug that has been used in the lab for a couple of decades. People think they're studying PXR activity, but they might be studying other nuclear receptor like PPR gamma's activity as well. So it's kind of a cautionary tale. It's not all that clinically relevant because SR is a failed drug candidate, but it broadens the knowledge base in general. And that's kind of what we're about. Okay, I'm going to wrap up the talk really quick here to highlight the future directions. So that was colon cancer. It turns out we have molecular biology magic tools, and this vector is one of them, in which we inserted the Pregnetin X receptor cDNA. But what I want you to notice is this zeaxanthin gene. This is a gene that expresses a protein that when you add the drug to the culture media, it kills all the cells that don't have this plasmid. And this plasmid carries a promoter, the cytomegalovirus promoter, that drives expression of PXR cDNA. So what you do briefly is you linearize this plasmid, transfect cells, and at some level it integrates stably into the chromosome. So we're making stable cell lines. And when you add the zeaxanthin, it kills all those lines that don't express zeaxanthin. And if they express zeaxanthin, they're likely to express your gene of interest, which kind of hitchhikes along on this plasmid. So Sanvi Kohli, a very bright, articulate, eager M-keeper, I call him, in my lab, was extremely interested, and she just kept showing up in the lab. So I put her to work, and she and I, with the help of Dr. Creamer and other medical students, created several different cell lines called MDA MB231 wild type, so not expressing PXR, are those that are PXR positive. In other words, they were resistant to the antibiotic zeaxanthin. So what is our gene expressed? So I asked Sanvi to come in on the weekend, and we actually did Western blotting using the antibody that recognizes anti-PXR. On the left, you'll see just a general protein stain of all the proteins isolated from these cell lines. So wild type, clone 1, clone 2, clone 3. So we had 3 independent clones all expressing resistance to zeaxanthin. The question was, do they express PXR? And Sanvi was able to show that clone 1, 2, and 3 do express PXR. So that was really exciting for her. And we'll have a poster both at KCU Research Week as well as MSSU Research Week on this data. So Sanvi, again, is a very bright M-keeper student. Appreciate M-keepers so much. And again, Dr. Rhodes, thank you for integrating with the laboratory as much as you have. Okay, why would we want to do this? Okay, it turns out certain nuclear receptor family members have this weird phenomenon, and it has to do way back when I told you about dexamethasone. It's anti-inflammatory. Well, it turns out many nuclear receptors are anti-inflammatory. PXR activates what I'd already described to you as the xenobiotic response, induction of drug-metabolizing, drug-transporting proteins. With respect to inflammation, we heard a little bit about it. TNF-alpha activates NF-kappa B to mediate the inflammatory response. But it turns out these two pathways cross-repress each other. Now, how does this, and they call it trans-repression. How does this manifest clinically? It turns out that morbidly obese, inflamed, metabolic syndrome, diabetes, cirrhosis, any kind of chronic inflammation of the gums increases the inflammatory response, which has this transrepressive effect on the xenobiotic response. So inflamed patients, future clinicians, tend to retain drugs in their body because of this phenomenon. So we're setting out to study that in the context of breast cancer, this transrepression phenomenon. So this summer, I'm going to engage with M-keepers and student doctors to ask the very fundamental question using those stable cell lines. Does expression of PXR in a triple-negative breast cancer cell line, which doesn't normally express PXR, does it have an impact on the inflammatory response, which has a lot to do with tumor aggressiveness, and so on and so forth. So we're on the cutting edge, and here's some of the folks. Sanvi is right here, and some other M-keepers here, and other student doctors, myself, Dan. So I'll stop there and take any questions, and thank you so much, you guys. And I need to be forthright and say, oh, I didn't mean to do that. National Cancer Institute recently gave us a R15 area award, so we're well funded at this point. We're looking forward. The interface between MSSU and KCU would be key to renewing this grant, training young healthcare professionals in the context of KCU, MSSU interface. So we're super excited about that. Any questions that I could answer? How'd I do on time?
You're doing great.
Oh, thank you.
Questions will come to you.
Did I hit the sweet spot? Was I over your head?
Okay. Thank you. Well done.
Were you bedazzled?
Thanks, Dr. Staudinger. I just want to say, wow, that's happening right here on the campus of Missouri Southern State University. Isn't that amazing? Am I the only one that's impressed by that? I'm pretty impressed. I don't think I knew that. So good job, Dr. Staudinger and team. So let's give another round of applause for all the speakers today. I think it's been a great day. We've had a really wonderful opportunity to learn from such knowledgeable national and international experts right here in Joplin, which doesn't happen very often. So thank you to Missouri Southern State University for making that happen. And for the final time, please scan the QR code on the screen to complete the form for your CE credits and class credits for attending this session. And then it is my pleasure to introduce Dr. Dean Van Galen again to come up to the stage and close out our summit.
Oh, it's been a great day here at Missouri Southern. Thank you all for being part of the second annual Wilcoxon Innovation and Health Sciences Summit. Our speakers were tremendous, gave us really some powerful insights into the future of health. Again, it's so great to see our students here, our faculty, our campus community, and broader community come out today. So I did want to express thanks, obviously, to our speakers, our sponsors, the planning committee that made all of this happen, certainly the Wilcoxon family. And I do want to close by announcing next year, of course, we'll have another summit here on the Missouri Southern campus. The Roy Blunt Health Science Innovation Center will not quite be done, but perhaps we could arrange some tours of that site during that time. The date for next year's summit is March 27th, 2026, so I hope you'll mark your calendars and join us again next year. Thanks for being here at Missouri Southern State University. Go Lions.